Hello, my name is Anna Fernazan. My topic is genetic engineering in vaccine development. The focus of my project is to explain how genetic engineering allows vaccine to be developed more rapidly and safely by using genetic instructions to produce antigens inside human cells and to evaluate the effectiveness, limitations, and ethical considerations of this technology. What are genetically engineered vaccines? Genetically engineered vaccines use recombinant DNA technology to improve the effectiveness, safety, and adaptability of vaccines. Instead of introducing whole pathogens into the body, these vaccines deliver genetic instructions such as DNA or mRNA, which allow human cells to produce a specific antigen. Because only the antigen is produced and not the entire virus, genetically engineered vaccines are considered safer and more targeted than traditional vaccines. The vaccines differences between traditional vaccines and genetically engineered vaccines. Traditional vaccines often rely on weakened or inactivated pathogens to stimulate an immune response. Although effective, this method can take longer to develop and may pose risk for immunocompromised individuals, as it still involves exposure to viral components. In contrast, genetically engineered vaccines use only DNA or mRNA, which instructs cells to produce one specific antigen. This allows the immune system to respond without exposure to the whole pathogen, making development faster and reducing the safety concerns. Immune response overview. When a pathogen enters the body, it produces proteins called antigen, which are recognized as foreign by the immune system. In genetically engineered vaccines, these antigens are produced inside whole cells. The antigen is first recognized by T lymphocyte, particularly helper T cells, which coordinate the immune response. These T cells then activate B lymphocytes, which produce specific antibodies that bind to the antigen. At the same time, memory B cells and memory T cells are formed, allowing a faster and stronger immune response upon further exposure to the same pathogen and makes a body stronger. The development of a genetically engineered vaccine begins with identifying the pathogen and selecting an antigen that can trigger immunity. The gene encoding this antigen is isolated and modified using genetic engineering techniques. This gene is then inserted into a delivery system such as DNA, mRNA, or a viral vector to create a vaccine that can safely deliver the antigen instructions into whole cells. Viral vector vaccines use a genetically modified adenovirus to deliver the antigen gene into whole cells. The adenovirus is replication defective, so it cannot cause disease. Once inside the cell, the gene is transcribed into mRNA and translated into a viral antigen protein. This antigen is recognized by T cells, which activate B cells to produce specific antibodies and immune memory. As a result, viral vector vaccines stimulate both cell-mediated and antibody-mediated immunity without infection. DNA vaccines involve the injection of a plasmid containing the antigen gene. After entering the host cell nucleus, the DNA is transcribed into mRNA. The mRNA then exits the nucleus and is translated by ribosomes into antigen proteins. These antigens activate immune cells and promote the formation of antibodies and immune memory. mRNA vaccines introduce synthetic mRNA directly into the cytoplasm of host cells. Unlike DNA vaccines, mRNA vaccines do not enter the nucleus, making the process faster and more efficient. The mRNA is translated into antigen proteins by ribosomes, which are then recognized by the immune system, stimulating antibody production and memory cell formation without using live pathogens. Research shows that genetically engineered vaccines can be developed much faster than traditional vaccines. During the COVID-19 pandemic, mRNA, DNA, and viral vector vaccines entered human clinical trials within months of the viral genome being published. These vaccines have been successfully used against diseases such as hepatitis B, COVID-19, HPV, and in veterinary medicine, demonstrating their effectiveness in real-world applications. Despite their advantages, genetically engineered vaccines have limitations. mRNA vaccines are less stable and often require cold storage, creating logistical challenges in distribution. Another limitation is immune escape, where rapidly mutating viruses may reduce vaccine effectiveness over time, requiring vaccine updates or booster doses. All genetically engineered vaccines undergo extensive safety testing, including animal studies and phase human clinical trials. These trials assess safety, immune response, dosage, and potential side effects. Regulatory agencies requires each genetically engineered vaccines to meet strict standards, ensuring it is safe and effective before approval for public use. Genetically engineered vaccines raise ethical considerations, particularly regarding long-term monitoring, cost, and equitable global access. However, their ability to be developed rapidly during disease outbreaks represent a major ethical advantage in protecting global health. In conclusion, based on current scientific evidence, genetically engineered vaccines represent a safe, effective, and ethically justified advancement in vaccination strategies when properly regulated and continuously monitored. Thank you very much for your time and attention for genetic engineering in vaccine development.